Our project focuses on what antigens are, what antibodies are, and how they bind.
Suppose that this is an antigen and this is an antibody. As you can see, the antibody doesn't fit with the antigen, meaning that the antibody won't have any effect. But if we look at two pieces that do fit together, the antibody binds to the antigen through this piece. Called the epitope, which allows the antibody to neutralize the pathogen and mark it to be destroyed by other cells.
Antigens are usually foreign and are typically large molecules, either proteins or polysaccharides. The small, accessible portion of an antigen that binds to an antigen receptor is called an epitope or antigenic determinant. An example is a group of amino acids in a particular protein. A single antigen usually has several different epitopes, each of which binds to a receptor with a different specificity. Because all antigen receptors are produced by a single B cell or T cell are identical, they bind to the same epitope. Each B or T cell thus displays specificity for a particular epitope, enabling it to respond to any pathogen that produces molecules containing the same epitope.
An antigen is any substance that elicits a response from a B or T cell. In adaptive immunity, recognition occurs when a B cell or T cell binds to an antigen, such as a bacterial or viral protein, via a protein called an antigen receptor. Antigen receptors are specific and bind to just one part of one molecule from a particular pathogen. There are about 100,000 antigen receptors on the surface of a single B or T cell. When infection by a virus, bacterium, or other pathogen occurs, B and T cells with antigen receptors specific to that pathogen are activated.
In this presentation, we have concluded that antibodies bind to antigens through epitopes.